Our next talk is from the same region of the world, also from Finland, PEKKA Postila, who's going to talk about modeling the effects of SYNGAP1 missense mutations. Thanks.
Yeah, thanks for the opportunity to talk about my research. So I'm going to talk about modeling structural effects of SYNGAP missense mutations using molecular dynamic simulations. So firstly, a little bit of words about the aims of the work, what we're doing. So the primary sort of aim, what we're working with, at least in kind of what I'm talking about in this talk is the assist in the diagnosis of these missense variants of unknown significance. But at the same time, obviously, when we're looking at the SYNGAP1 structure, protein structure, we want to see some sort of connection between the function and dysfunction. Kind of increase the understanding of that. And then finally, obviously, we would like to participate somehow on a structure-based drug discovery for SYNGAP. So, a few words about the structure. Here's a rough sketch of the different domains that are included in SYNGAP. As you can see from Michael's presentation, there are a little bit more information about this, but the main ones, I guess, for my purposes, is the pH domain and C2 domain. They are interacting with the membrane, possibly. I would claim that the C2 domain is the part that actually interacts with the membrane. Then there's the GAP domain, which is the GTPase activating protein part. So there's the R-gene finger, which is interacting with the GTPase, for instance. And then there's the coil-coil domain, where the trimerization supposedly happens. So if you actually look at the structures, the experimental structures that are available, there's actually a lot of stuff missing from that protein. And so we have the coil-coil trimer, and then we have sort of a fragment of the C2 GAP domain together. So because the situation is that, we'll have to use AlphaFold. Fold2 luckily has a model for this. And well, if you look at it, it looks quite kind of messy, stringy. But if you actually take away the parts that are unreliable, even by their standards, then you end up having this type of model, which has most of the pH domain, and it has the C2 domain, and there's the RAS-GAP domain as well, in complete size. We'll also include it like sort of a disordered area, which is in the C2 domain, this glycine-rich loop, which in the simulations is actually interacting with the membrane. So just to show that the model is not like a figment of imagination, it actually is based on the structures. So you can see here with this light cyan color, the AlphaFold model. And if you compare it to these crystal structures with the what is available there, it's almost a perfect match for most of the structure. And there's the C2 domain. I'm not 100% sure where it got this part, but there's plenty structures for the C2 domain as well that is probably used for this. So it's sort of like a homology model. You can think of it that way. So when we use this sort of N-terminal model in the simulations, we have 210 variants at the moment. It's growing the number all the time, but so we can, is the mic still working here? So we can basically study these missense mutations with the structure, and there's plenty of them. You can see here the coloring for the assigned variants and then the unknown ones. So a little bit words about the methods. I won't go into too much details, but basically we do in silico mutagenesis. So we use the computers to generate the missense mutants using a software called Modeler. We've tried also AlphaFold. You can also generate them the same way, but we get similar kind of structures using that. And yeah, so now we get the mutation done for the structure, and then we run an MD simulation. There should be a movie moving around showing this with solvent simulation. So we just put the protein in the solvent, and we simulate it using atomistic simulations. So we get this thermal motion of the protein, how it interacts, well, kind of what the dynamics are. So we do this in 3 replicas with 150 nanosecond simulations. and for all of the variants. We also do calculations with the RAS, SYNGAP complex, and with membrane. But I'm just going to focus on the solvent simulations in this talk. So although 1 nanosecond is like 1/1,000,000th of a second, there's still quite a lot of computing to do with this method. So we have to use supercomputers for this. And luckily in Finland we have CSC, which is providing us really nice resources to do this type of work. The amount of data is really huge, and actually running the simulations faster than performing the analysis. So what can you expect? What is the balance view of this? So there's a lot of pros into this. So you can actually see what the mutations, where they're located, what they're sort of doing. So that's better than looking at the sequence. We can see definitely local changes in the stability and the conformation of the protein from the simulations. And in many cases also we can see like what is benign, what is pathogenic. It becomes obvious once you see it. The bad thing or the con to this would be we're actually working in a sort of backwards manner, because we have the fully folded protein and we're introducing the mutations at that stage. So if the effect is felt during the folding, we're not necessarily going to see that. And we're not going to see complete unfolding of the protein either, even if that would happen, although because of how the approach is set. So a couple words about the results. So we've tried different ways to categorize these mutations, what they're doing. So if we look at the structure. So here's a bunch of missense mutations that are affecting the secondary structure, located in secondary structure elements. Then we have some of the residues are buried, or the side chains are buried, so when the exchange happens, it's inside the protein, the side chain. And then here's a bunch of mutations that are close to the membrane interface, so they might affect the membrane association. And here's a bunch of mutations also at the interface with the RAS, GTPase. And then a bunch of these mutations are on the surface of the protein, and they could be more likely to be benign because of this, because they're not necessarily having other effects. But these kind of overlap these categories like this. So we've tried to come up with some sort of shortcut, because there's so many of these, and when you look at the structures, that we could actually see, what makes sense, what would be pathogenic, what would not. We haven't really found any metric like that from the simulations so far. So that's a problem. But at the same time, I have to say, like, if you actually carefully look at the structures, you start to see clear signs there. So if there's an effect, then it is potentially pathogenic, and the opposite with the benigns. So firstly, we started our work with these 3 variants. So I'm going to talk about a little bit of these, so you can see the effects in the simulations. So the first example is this leucine 323 proline variant. And in this case, it has an effect on the secondary structure, so it kind of disrupts it. So in the wild-type structure, we have leucine there, and leucine has a backbone amino group which is able to participate in this strand formation, beta strand formation there. And when it's exchanged for proline, it doesn't have that group in the amino acid, and it has a clear effect on the protein structure. And there's actually plenty of these type of mutants or variants that are known. Another example is actually the same residue, which is swapped for arginine in this case. So the side chain is actually buried in a hydrophobic sort of niche in the protein. And when it's exchanged to arginine, it introduces a positive charge or residue inside the protein. And as you can see here, from the magenta to the yellow part of the structure, there's a big effect in the structure. And it's even more kind of obvious once you actually look at it close by. So the arginine has to move out of the pocket so that it's too polar for that, or charged for that space. And you get this major adjustment. Another example here is a glycine 344 serine variant, and basically here the mutation happens really close to the membrane, or where we think the membrane is, And once this exchange happens, now it starts to actually affect the loop dynamics. So glycine doesn't have a side chain at all, basically. It only has a hydrogen there, but serine has a hydrogen group, so it's able to make hydrogen bonds with nearby residues. And and also with lipids, and that will affect the membrane association. So what are the sort of conclusions? So actually the AlphaFold model is actually pretty robust under simulations once you use the reliable parts of it. And we see these local changes for sure. We can even get it to a little bit unfold. We just boil it, with 400 Kelvin temperature. The most of the movement happens with the pH domain, which was not fully kind of included in the model. With some mutations, it's not really working that well because basically we're introducing similar sized residue somewhere inside the protein, and the force field does its best, but it's not able to necessarily fix it. And also I want to say that the molecular dynamic simulations, they're not really a magic bullet, so you can't expect them to provide everything you need, but basically you get the structural perspective as opposed to the sequence-based analysis, and there's no metric that will just magically tell everything, but if you look at the structures carefully, you actually can gain information out of these simulations. And for distributing the data, we're actually building have already kind of built a server, which is in the final stages, called Syngap missense server. So I'm just going to show a few screenshots from the server. I kind of borrowed the colors from the SRF here for the server, but let's see what it's going to finally look like. So it's a website. You can go there with any browser and just look at the Syngap data. Well, mainly our data now, but also other stuff. Free and downloadable. The coding for the 3D viewer is going to be done, or is done with Molstar, the same as with PDP. And it's still under development, so I'm not opening it just yet. But if you're interested, you can, ask after this event, and I can show you how it works. What it contains, it has these in silico models, variants what we made from simulations. There's also sequence and machine learning and physics-based servers analysis, which is done. And there's clean-one data, kind of, at least the status is included, but we could include other stuff as well. This is the landing page, at least at the moment. And here's the 3D viewer. Obviously more practical when you actually use the mouse and look at it, but what it has is, like, you can choose any variant, or you can search for it. There's a list there. You can go through different kind of models from the MD simulations from 0 to 150 nanoseconds, so you can see the change, what happens. You can even get the membrane plane showing up, so if you want to see where the membrane would be. And in addition, we also have a data table included for each of these variants. So there's a bunch of analyses included. And this analysis includes the sequence-based metrics, what we calculated, and also the structural annotation, what we've seen happening in the due to the mutations in the structure. I guess the major part is this written description, which we're going to include for each of these variants, where we actually explain what we see in the simulation. So that should make it easier to look at the structures yourself. On top of that, it'll have data plots as well that you can look at from the simulations. So a couple words about the sponsors. So obviously Syngap Research Fund and CSC and Inflames, those are the major contributors. And here are the people involved in the project. So Professor Olli Pentikäinen, and is co-supervising together with me the lady in the fur coat, Alia, and my PhD student. And then there's Henna-Riikka doing some sequence work. Obviously Michael that you already saw, and Lily, and Jukka Lehtinen on the far right. He's doing the coding for the server. Was I fast? Yeah.